Alright guys, welcome back to Underground Science, and in this video we're going to be talking about the basics of DNA. So let's start off by saying that DNA is located inside the nucleus of the cell, and we know that DNA has a sugar phosphate backbone. Specifically, that sugar is a deoxyribose sugar because we're talking about DNA, and we know that because on the second carbon here we see only an H and not an OH. If we saw an OH, that would be a ribose sugar, and that would be RNA. Speaking of RNA, we should remember that in RNA, we do not have thymine as a nitrogenous base. Instead, we have uracil, okay? So that's good to keep in mind. So we can see here that DNA is arranged in an anti-parallel fashion. That means at one side, we see it starts off at the 5' phosphate end, and then it points towards a 3' OH end. But on the other side, right, this side, we see it starts at the 3' end, and then ends and the 5' phosphate end. So that's why DNA is said to be arranged in an anti-parallel fashion. Okay, so knowing that, let's move on to talking about the fact that, there's the difference between purines and pyrimidines. So purines are, the nitrogenous bases that make up purines are adenine and guanine, okay? They're 2 rings each. Pyrimidines are cytosine, thymine, and uracil. They're only 1 ring each, okay? And something to note here is that adenine and thymine, they have 2 hydrogen bonds and they bind together. And then cytosine and guanine are held together by 3 hydrogen bonds and they bind together. I feel like it's also important to note that for future, like when we talk about histones, is that the sugar phosphate backbone that we see is actually negatively charged. And the reason for that is because of these phosphates, okay? These phosphates are negatively charged, so that makes the whole sugar phosphate backbone negatively charged. Okay, and let's just keep that in mind for now. So I feel like an important distinction to make here is that we have to know what is a nucleoside and what's a nucleotide. Okay, let's make that distinction. So really quick, a nucleotide is the sugar, the phosphate, and the nitrogenous base. Okay, so for example, if we zoomed in here, I'm talking about the sugar, the phosphate, and the nitrogenous base. So this whole thing right here with the sugar, phosphate, and nitrogenous base is a nucleotide, okay? So that's what DNA is. We're basically putting a bunch of, like in DNA replication, we'll see you're just joining many nucleotides together, right? And so that's a nucleotide, but a nucleoside is what we just said, except just take out the phosphate. So it'd be a sugar and nitrogenous base, but no phosphate, okay? That's nucleoside. So that's the difference between a nucleotide and a nucleoside. So I have some examples here. So let's say we take into consideration adenosine. Okay, let's take adenosine first. Adenosine is a nucleoside. Now, why is it a nucleoside? Because it's made up of a nitrogenous base right here. Let's get that pointer. Nitrogenous base and a ribose sugar. So remember we said base plus sugar equals nucleoside. Same thing here. Thymidine, right, is a nucleoside. Why? Because it's made up of a thymine nitrogenous base and a ribose sugar. So base plus sugar, nucleoside. Same thing with uridine and cytidine, right? We have uracil and sugar makes up uridine nucleoside. Cytidine, we have cytosine and sugar makes up a nucleoside. So base plus sugar. And so if we keep on doing that, now I think we understand that a base plus a sugar makes a nucleoside. Okay, and specifically these are RNA molecules because there's ribose sugars that make up these nucleosides. If this was deoxyribose sugars, we would have DNA. But so if we keep on going down, right, over here we were supposed to see guanosine, but let's just write that in since we don't see it here. Okay, so again, to make this even more ingrained in our minds, again, we see here, guanosine is a nucleoside. Why? Because it's made up of a guanine nitrogenous base and a ribose sugar, which makes up a nucleoside. So if we come down here and we talk about deoxythymidine, right? Just taking one example. So the only difference between thymidine and deoxythymidine is that now we're talking about DNA, okay? We're talking about DNA now. So let's go and erase this since this decided to show up. Sorry about that. All right. So again, the only difference between deoxythymidine and thymidine is that deoxythymidine is DNA. So we're taking, again, we are taking thymine, nitrogenous base, combining that with a deoxyribose sugar to make, again, it's still a nucleoside because it's nitrogenous base plus sugar, but it's DNA this time. All right. So one last thing that we have, or we should know about this is that when we're talking about nucleosides versus nucleotides, so what's an example? So I'm sure we've heard of AMP and ATP, in many metabolic processes in biochem, like in the Krebs cycle or glycolysis and many others. So adenosine is a nucleoside. Why? Because it's made of adenine and a ribose sugar. But if you add a phosphate on that, you make it a nucleotide, which an example would be like adenosine monophosphate, which is a nucleotide, or adenosine triphosphate, which is basically adenine, ribose sugar, and 3 phosphates. All right? So now that we know the distinction between these, let's go on to the next page where it's going to be important to talk about histones. So histones are basically made up of 4 proteins, okay? So here, let's say this. So a histone protein, okay, has 4 individual parts to it. So what are those 4 parts? So we have an H2A, okay? So instead of that, let's just use a pointer. We have H2A, H2B, H3, and H4. So that's 4 parts, right, to the histone. However, we have 2 of each. So that's why we call it a histone octamer, because it's, 8 individual subunits. So a histone octamer actually has DNA wrapping around it twice. So the DNA wrapping around the histone octamer, okay, right here, you see how this DNA circles around it twice? So the DNA that circles around it twice plus the histone octamer, that's known as a nucleosome complex, okay? So it's a nucleosome. Again, that's when you have your histone octamer, which is made up of 2 of each, right, of H2A, H2B, H3, H4. That histone octamer has DNA wrapped around it twice. So each individual complex like that is called a nucleosome. Okay, and another thing to know is that there's also another part of a histone. It's called H1 histone protein, and that's basically, it binds onto the nucleosome, right, and onto this DNA. You see this DNA that basically links one nucleosome to the other. So that's why this is the H1 histone, because it literally binds onto the linker DNA, which is the DNA that links one nucleosome to the other nucleosome. And this, specifically the H1 protein, binds onto the nucleosome and onto the linker DNA, okay? So it's good for, again, linking both nucleosomes together. So knowing that, let's talk about histones, right? More about histones. So what are histones? Like we said that DNA is wrapped around the histones, right, to form the nucleosome complex. But DNA is negatively charged. So how does histone bind onto that? That's because histones are positively charged because they have 2 main amino acids, lysine and arginine, and we know that lysine and arginine are positively charged. Okay, so let's write that down. Lysine and arginine are positively charged, which means that positively charged things, right, like negatively charged things. So opposites attract. So that's why histones can very easily bind with DNA, because DNA, again, is negatively charged. Okay, so knowing that, let's talk about the differences between heterochromatin and euchromatin. So the only difference that we should know is that heterochromatin is condensed. Okay, you can't really access the DNA, so you can't express it. So you're not going to be able to do transcription on it if it's heterochromatin. And one of the reasons we'll talk about later is because heterochromatin is usually methylated, meaning if you methylate something, it basically reduces the expression of that certain genetic material. Okay, but we'll talk about that soon. So All we have to know is that heterochromatin, you cannot access DNA, so it's condensed. Because it's condensed, you can't access it. So for example, right, if you look here, we zoom in here, you look right here, notice how the histone and DNA are tightly interacting with one another. That's because this DNA is heterochromatin, right? You can't access it because of the tight interaction between the histone and the DNA. So you're not going to really express that gene. However, if we take euchromatin, that's loose, okay? So that's a loose connection between the DNA and the histones. That means you can access the DNA. So if we look here, right, look at this portion right here. Do you see a tight interaction between this and a histone protein? We don't see that. That's because that's euchromatin. So that means DNA is easily accessible. We can go there and express that gene, right? So if we have to do transcription translation, we can do that. As compared to here, it's such a tight interaction with the histone, like no enzymes can really come there and start its transcription process. That's why we say heterochromatin is not, you can't access the DNA, but in euchromatin, you can, okay? So some epigenetic factors and concepts that we should know is DNA methylation is basically if you see some like cytosine guanine, right? If you see a bunch of cytosine guanine somewhere or CpG islands, just know that that can get methylated, okay? And that's a methyl group. It can get these CpG islands can get methylated. And remember, if you get methylated, you reduce gene expression, okay? Just like in heterochromatin. If you methylate CpG islands on a DNA, like let's say there's a CpG island on a promoter, if you methylate that, that gene will no longer be expressed. Okay, so methylation decreases gene expression. Okay, decreases or even silences the gene, so you can't even express it. Histone, then there's histone acetylation. Okay, so there's methylation, then acetylation. If you take a histone protein, remember, histone proteins have lysine and arginine, so they're positively charged, right? If you take histone proteins and you acetylate it, you add acetyl groups, you literally loosen the interaction between the histone and the DNA. Okay, you make the histone proteins less positively charged, therefore they interact with the DNA much more like loosely. Okay, they're not as, I should say, they're not a strong interaction anymore between the histone and DNA. Since you reduce the positive charge of the histones won't really interact that tightly with the DNA anymore. So you're technically making euchromatin, right? You're making a loose interaction between the histones and the DNA, which means acetylation increases gene expression. Okay, so methylation decreases gene expression, acetylation increases gene expression. All right. Well, before we move on, something cool to note that we'll talk about later when we talk about dive more into genetics and its diseases, associated diseases, is that Fragile X syndrome is a condition that's associated with DNA methylation. Sometimes this process can get messed up in certain individuals, which can lead to Fragile X syndrome, but we'll talk about that later. It's just a good clinical association for now. Let's move on to histone deacetylation. Okay, so if we understand histone acetylation, right, you add acetyl groups to histones, that leads to a loose connection now between the histone and DNA, which frees the DNA for enzymes that are involved in transcription and translation, right? Specifically transcription to start gene expression, and then you can express your genes. So acetylation increases gene expression. That means deacetylation means you're taking out the acetyl groups from the histone, or you're taking the acetyl groups off the histone proteins. That means you're just doing the opposite. So if acetyl groups increase gene expression, if you take out the acetyl groups from the histones, right, that's going to cause a tight interaction between histones and DNA. So heterochromatin. Okay, because now, remember, acetylation decreased the positive charge on the histones, like it decreased the interaction between the positively charged histones and the negatively charged DNA. Acetylation did that. But if you take those acetyl groups out, now you have a strong interaction again between the positively charged histones and the negatively charged DNA. Okay? So knowing that, we now know that histone deacetylation is related to decreased gene expression, okay? Because now you form heterochromatin if you deacetylate something. And another fun thing to know that we'll talk about later, by the way, when we talk about Huntington's disease in more detail, is that sometimes there's some proteins that you need to make, right? Like for relating to the, let's say the basal ganglia. And again, we'll talk about this later, but let's say you can't make those proteins that are needed for the basal ganglia, for example. And if you can't make those proteins because you deacetylated a lot of your histones, that means you have a tight interaction, heterochromatin, between the histones and DNA. That means you cannot express your genes, means you cannot do transcription and further make proteins. That can lead to Huntington's disease. All right, and we'll talk about this further when we actually dive more into Huntington's disease itself. All right, so I feel like that was a good start on the basics of DNA, and it'll help us later when we talk about purine metabolism, pyrimidine metabolism, and so on. All right, so thank you guys for watching, and I see you guys next time.